Hello, and welcome to the So You Want to Be a Marine Biologist podcast, where we learn about the ocean, share sea stories, and explore ocean careers. I'm your host, Kara Muzia. Let's dive into today's episode. Today's guest is Gabby Barbarite, FAU's Harbor Branch Oceanographic Institute's Director of Outreach and Engagement. Growing up in South Florida, Gabby was always called to the sea. In fact, during high school, she earned her dive master's certificate and quickly became a dive instructor. She found that though she loved diving with people and explaining the oceanic denizens, she herself wanted to learn more. So Gabby enrolled at Florida Atlantic University and found a calling in the shark lab. She decided to continue her schooling all the way up to her PhD, and along the way, life threw a couple of twists. Incidentally, this is when I met Gabby. She was actually my TA or teacher's assistant in one of my classes at Harbor Branch. In this episode, we chat about diving in submersibles, what to do when you have to change your PhD thesis a year or two into the game, and all about Vibrio vulnificus, or flesh-eating bacteria. Here's Gabby. Gabby, welcome to the show.
Thank you for having me.
So I kind of want to jump right in with you. We have a lot to talk about. Your research and your career kind of has covered so much. So first off, I want to go back to the beginning. How did you get into marine biology, and was there a defining moment?
Ooh, that's a great question. So I was born and raised in South Florida, and so I fell in love with the ocean at a very early age. I grew up, snorkeling, and as soon as I was old enough, I got scuba certified and spent lots of time in the water. And I worked as a dive master and a scuba instructor for many years. And I always loved telling people about the ocean and about all the cool creatures that we'd see on our dives. And the more I did that, the more I realized how awesome these environments were and how much I wanted to share them with people.
Did you graduate high school and then decide to become a dive master?
I did. Actually, while I was in high school, I went for 2 summers to the British Virgin Islands and worked on a sailboat as a dive master. And so I actually got certified one summer and then I went back to work for the next summer. And so that's when I really just decided that I loved being in the water and teaching people about the ocean.
I feel like that's like a dream situation. I'm going to go live on a sailboat, and then I'm going to teach people, I'm going to dive, and I'm going to teach people how to dive and about the ocean. That's really awesome.
It was super cool.
Okay, so you decided wrap up dive masters, and that kind of like piqued your interest in marine biology as a career. So did you come back stateside and go back to school?
I did. I started my bachelor's degree at Florida Atlantic University. And while I was doing that, I decided to volunteer. And so I worked with the Shark Research Lab there. And so, of course, like any student, you have to start out washing glassware and cutting up little scraps of fish to feed, or things to feed the fish, and slowly became more interested in research and got to do more hands-on things.
Was that in Dr. Kajuro's lab?
It was.
So that's just completely during your undergrad, and you kind of fell in love with sharks. Is that kind of how that panned out?
Yeah, that was my original interest. I wanted to do shark research, and so that was the perfect lab to be in. But then when I was a senior, I heard about Harbor Branch Oceanographic Institute, which is an FAU campus that's up in Fort Pierce, so about an hour and a half north of the main FAU campus in Boca. And they have a program called Semester by the Sea, and it's for students at any university. Actually, they've just recently opened it up to any students, so you don't have to be at FAU. And you can come and spend an entire spring semester here, and all of your marine science courses are taught by the researchers at the Institute. And so I came up for that, and it was just amazing to see all the different fields of research and to get to do all these hands-on, pretty complicated labs and get to go in the field. And it was really eye-opening just how many different possibilities there are in marine science.
There truly is. So what was some of your favorite classes while you were at Harbor Branch?
Ooh, that's a good question too. There's a class called Marine Biodiversity. I think that's probably one of the favorites among most of the students that take it. You basically go through the entire taxonomic tree of all of these marine organisms. You start at algae and you end in marine mammals, and every week is a different lab. And you get to go out in the field and either collect or observe the animals. And so you go seining for fish and do plankton tows and go bird and dolphin watching. It's just super cool.
I know I took that class during my time at Harbor Branch, and it was that the one that like there was four different teachers that cycled through?
Yes.
Yeah, that was a really incredible class.
Makes it interesting. And then the other one, did you take the research cruise too?
Yes.
Yeah. So there's another course called the Oceanographic Experience, and you basically go on a research cruise, which is so cool as an undergraduate. And you go out to sea. Now they're running them in the Gulf of Mexico, so you leave out of Tampa, and you spend 2 days out at sea, and you get to collect all these different animals and identify them. And then the students actually do a research project and present a poster like a month later. It's a very quick class to all the researchers at Harbor Branch. So that was a really cool thing too.
Yeah, it was a really good experience to see truly from start to finish in a very abbreviated manner of, what you could possibly do research on, and to actually go from data collection to analysis and all the statistical analysis that comes with that, and then presenting a poster in front of, your colleagues and your professors. So that was a really unique experience for sure.
Yeah.
So you did Harbor Branch, Semester by the Sea, and just fell even more in love with marine biology. And from there, did you get your master's?
I did. So I actually, well, between those things, Harbor Branch also has a summer internship program. And so
Okay.
participated in that too. Worked with Dr. Brian LaPointe on harmful algal bloom research. We were looking at algae that were growing over coral reefs in Florida and kind of smothering out some of the corals and the sponges. And so we did some research on that. And then for there, I decided to go for graduate school. I knew it was something that I wanted to do for a long time. And I actually, for my PhD research, I wound up working in a microbiology lab here, which again was not something I had ever really thought of doing. My project was actually really cool. So I studied a type of bacteria called Vibrio vulnificus, and a lot of people don't know of that bacterium, but you may have heard of the flesh-eating bacteria that's been at the beaches in Florida.
Yes.
So every year, Florida has the highest number of these infections, and it's a pretty serious hazard if you do become infected. However, no one's monitoring these bacteria in the environment. And so we set out to do the first ever study looking at these bacteria in the water and figuring out how people can become infected.
Hey, Kara here. I hear it all the time. How do I become a marine biologist? Or my niece wants to help the ocean. Where does she even start? I hear from students, yes, and also nurses, HR professionals, and other mid-career folks who wonder if they could still make their dream of working with sea turtles or sharks come true, wondering if it's too late to answer the ocean's call. Here's what I've learned after 20 years in the field and hundreds of interviews. There's no straight line, no magic degree, and no gatekeeper. You create the path forward. If this sounds overwhelming and vague, I've created 2 tools to help you stop wondering what if and start moving forward with clarity and purpose. The first is my free guide, Be a Marine Biologist Without the Degree. Over 1,000 people have already downloaded this guide and taken their first step towards ocean impact. Inside, you'll find 7 concrete, doable actions to help you get started, whether you're just exploring or ready to make a change. Then there's the Ocean Blueprint. This is a 30-day self-paced course that helps you build your unique path into ocean science and conservation without quitting your job or going back to school. Inside the Ocean Blueprint, you'll get daily prompts to explore your interests, skills, and next steps, a structure that turns overwhelm into real momentum, and a bonus section to root your work into ocean connection. You don't have to wait for a degree, a perfect plan, or permission. Whether you're wading in or ready to take the deep dive, these tools are here to help you move from dreaming to doing. The ocean called. Are you ready to answer? Links in the show notes and on my website, karagracemuzia.com. I can't wait to see the waves you'll make. Okay, so what are the primary avenues of getting infected?
Well, that's a really great question. So they have the name flesh-eating bacteria, but that's not actually true. It's not like sticking your hand in acid and it's going to corrode your flesh. The way that you become infected is either you have an open cut or an open wound and you stick it in water where these bacteria are found, or you consume them, like from eating raw oysters.
Okay. So you can only get it from eating raw things, or if you have literally a raw cut exposed to the environment.
Exactly.
Okay. So are they found in all fish tissues, or is it just primarily filter feeders? Did you kind of look at that?
For eating them from getting sick from seafood, you mean?
Mm-hmm.
That's a great question too. So this is a side project that one of my students worked on.
Okay.
So we know that filter feeders like oysters accumulate the bacteria, right? Because they're filtering lots of water.
Right.
But and we know from my research, which I can talk more about in a minute, that these bacteria are found on the surface of fish bodies. And so if you're an angler and you get a cut on your hand and you grab a fish, you directly contact it. But people always wonder, is it actually in the muscle tissue of the fish? And so we did a study where we filleted the fish and we used sterile techniques to remove that skin layer. And we found that it wasn't in the meat if you did everything in a sterile way. Now, if you use the same knife to cut through the fish and then cut off that skin layer, or you took your fillet and you slapped it back on the cutting board, you could basically re-inoculate that tissue.
That makes sense. But who actually sterilizes their knife between, cutting the skin off and then cutting the meat? So
no one.
Yeah, right. So don't eat the sushi. You need to cook it first. And cooking it completely kills the bacteria?
Completely kills it.
Okay. So what else did your research uncover in your PhD?
So my main focus was trying to figure out how people can become infected. And so the first thing that we set out to do, since no one had ever looked for these bacteria on the east coast of Florida, is we sampled water and sediment from all around the Indian River Lagoon, which is an estuary. And we looked at if there were any differences in location on where these bacteria are found, and then also if there was a difference in seasonality. In other areas where these bacteria were found, say like in the northeastern part of the United States, they disappear in the winter because they can't tolerate cold temperatures. Florida, it's summer year-round, basically. And so sure enough, we found that they're present year-round in our waterways. And another thing that's important to note is that they are naturally occurring bacteria. So they've always been here and they always will be, and pollution isn't actually introducing them into the environment. So it's a tricky thing to control. But the other thing that we found was that it is related to the salinity, their abundance is related to the salinity of the water. And so they like brackish waters around 10 to 20 parts per thousand. And they're not actually found in the ocean, contrary to probably all the headlines that anyone's ever read about them.
Right.
They're not at the beach. You're more likely to encounter them in the lagoon next to a freshwater discharge source.
Interesting. So if it's too salty, that will kind of kill the bacteria, and similarly, they can't live in any fresh water.
Exactly.
Has there just been an uptick in encounters? Like, why is this, I've seen this more recently in the news more and more. Is this just kind of always been about the same levels? And I realize that you just said that nobody's really studied it before, but I feel like it's kind of become more prevalent recently. And is that just because bacteria levels might be up, or is that just because the news kind of found it and that's what we're seeing?
I think it's a lot of different things. So cases in emergency rooms have been reported since the 80s. We've had pretty good reporting since then. Um,
Okay.
as reporting methods get better, cases tend to also increase because they're getting logged. Um,
Okay.
another thing is that once the media, just like you said, their attention is turned on to a topic, they're looking for these stories and you're hearing more about them.
Okay.
But I certainly think that, and it's hard to say without having a baseline because no one has done these before, but I would say that Kate, that their abundance in the environment is likely increasing as we have warmer and warmer weather, more and more fresh water discharges and pollution into some of our estuaries. We're making these environments a little bit more favorable for them to exist. And so studies, 10 years from now are going to be really cool because we can look back and see what they were, when I did my research and how they've changed.
So from the 80s, we have data from the 80s that it's been reported. Has that kind of been the same from year to year? You kind of have the same amount of reportings from the hospitals each year, or that's also increased with the discharges?
I think maybe in the last 10 years, we've seen a slight increase. But another part of this research, so we looked for where they were and how they
Mhm.
might change over time and seasonally. And
Mhm.
then the real interesting part, and what got me to go into this project in the first place, being a shark person originally, um,
Mhm.
we wanted to look at fish. And so fish carry these bacteria on their skin. So just
Mhm.
like we have bacteria and microbes that live on our skin. They're not actually hurting us, they're just part of our natural microflora. And the same is true for fish carrying these Vibrio bacteria. And
Okay.
so we set out to do a survey and caught 600 individual fish one at a time. So we couldn't,
Wow. A hook and line, just one by one.
yep. So we couldn't use a cast net or a seine net because we couldn't disrupt the mucus layer
Right.
in the microbiome.
Okay.
So we had to do it one at a time.
That's
And
impressive.
we would get
Bet
the
you
fish.
got some really good fishing tips then.
Yeah, I learned how to fish. I never knew how to fish before this project. And by the end, I was like tying knots and casting and all these things,
Oh, that's so fantastic. The things you learn.
right? Pretty easy. I know we still get like, older guys would come up to me while I was fishing and they would say, like, oh, do you know this? And I'm like, yeah, actually, there's this living there and this living there. And last month we caught 12 of this.
I've had a lot of experience fishing lately. I've caught 600 fish.
Yep.
That's amazing. Okay, so you're out fishing and you catch 600 fish and you analyze each one. Was that just involving like you took a swab and kind of took a sample of their mucus layer on their skin?
Exactly. So we take two samples. The first one was a swab down the side of their body, and that kind of simulated if you have a cut on your hand and you touched it, that's what you'd encounter.
Okay.
And then the second sample, we took a fin clip. And so that kind of simulated if you got poked by one of the fin rays, what you might encounter in like your tissues.
Okay. And a fin clip is usually done with just like a really small, it's kind of like a hole punch, but smaller,
Yeah,
right?
we just used scissors and we just snipped off, like a centimeter of the fin. So really
Okay.
clean tissue sample. The good thing about microbiology is you don't need a lot of samples.
You're working on the micro layer.
We would bring all those back to the lab and we would grow them up overnight to see what would grow. And then we would test each fish to see if they were positive or negative for this Vibrio vulnificus.
What did you find? How many of these 600 fish had the Vibrio?
A really great question. So it depends on where you're fishing. And so
Hmm.
we tested two different sites. One was Harbor Branch, that was our control. And so it's an area that is not very developed. It's a natural shoreline, no major freshwater sources. The salinity was usually above 25 parts per thousand, so likely out of the range of Vibrio. And sure enough, they're rarely recovered here, except for 1 month where we had a ton of rain, and then almost all the fish here were positive.
Okay.
Now, compare that to our other site was right at the base of the Indian River main canal, which is the main drainage canal through the city of Vero Beach. And there, the salinity was anywhere between 5 and 15 parts per thousand, so right in their range. And sure enough, almost every single fish that we caught there was positive.
Wow.
So it really depends where you are if you're likely to encounter these bacteria.
I know you mentioned earlier that it's on their skin like we have microflora on our own skin, but is it truly either beneficial or benign on their skin, or is it detrimental to fish? Do we know that?
Some species of Vibrio can infect fish. Vibrio vulnificus often doesn't. That one's more of a human pathogen. There are about 100 different species of Vibrio bacteria. Vibrio is a genus.
Mm-hmm.
And one that you might be familiar with, Vibrio cholerae, is the causative agent of cholera. And so, again,
Okay.
major human pathogen. But in aquaculture, a lot of times they have problems with Vibrios causing infections in their fish. So it really just depends on the species. But this particular one that infects humans, and for the most part, the ones that infect humans don't typically infect other marine animals.
Interesting. So you can tell the difference between the species of Vibrio underneath a microscope. What is there like, one or two defining characteristics that separate them out, or is it require like more analysis than that?
Well, the great thing about microbiology is that they have all sorts of different types of media. And so, what we would do is we would plate out our samples onto a type of media called Chromagar Vibrio, and each different species would grow a different color. And
Mm,
easily count colonies and tell how many of each type were there.
okay. So what was the color of your species?
It was a beautiful teal.
All right.
Yep,
So anytime you had a teal slide show up, you knew that was your man, and that's what you counted. Interesting.
exactly.
That's really fun. That's really nice that you didn't have to sit there underneath a microscope and try to.
Try to count them. They're super tiny.
Yeah, that would be really difficult. Okay. Well, good. Yay, media and different cultures.
Yeah, our microbiology is just so amazing. There are so many cool things that microbiologists do, and all these different tools and types of materials that they use. It's really fascinating when you think about it, just how tiny microbes are and how they can detect them. And still, only about 3% of all bacteria in the world can be cultured. So there's still so many things that we hardly know anything about.
Oh my gosh. It's kind of nuts to think about.
I know the thing that always gets guests when I'm telling them about, like the microflora on these fish and on their bodies, it's like just like we have on our bodies. We have like 5 pounds of bacteria in our bodies, and people always, you see them like start itching.
Right. They're like all of a sudden my scalp, I need to itch it. I
Bacteria
mean,
are good.
not everybody realizes that we are our own biomes.
True. And so,
So where was your research published? I mean, I saw you in the newspaper quite a bit, kind of spreading the word about Vibrio and how to prevent getting infected. Where else can your research be found?
yeah, well, that was probably, in my opinion, the coolest part of my study. So, the more we were doing this research, the more we were realizing that no one here understood these issues, or were aware that these bacteria were out there. And so I made a pretty conscious effort to try to get that information to the public. And so when I first started my work, a fisherman actually died from being infected up in Melbourne, which is just a little bit north of us. And I remember reading the newspaper and seeing that his wife was quoted in saying that he'd been fishing in the lagoon his entire life and they never once heard about these bacteria. And
Mm.
so, as an effort to try to tell people what we were finding, we worked with a lot of local news agencies. So, in the beginning, I started out working with reporters and consulting on their stories. Toward the end of my time as a student, I actually wrote a couple columns, guest columns in the paper to try to get the message out. And then we also worked with a lot of agencies to try to produce like fact sheets. I had a fact sheet published through Florida Sea Grant, which is
Mm.
really cool. So it can be found online as well. And then we worked with the health department and then other medical agencies to try to make sure that the people that were treating these cases and receiving people that come in with these cuts and injuries are aware of these bacteria and what to do.
So well, prevention is to not go in the water with an open cut and do not eat raw animal essentially from the brackish water bodies where Vibrio is prolific, correct?
Exactly.
So if you do go in and you have an open cut and may or may not be infected, what, if any, steps can you take?
Well, the good thing about these bacteria, I guess if there is a good thing about such bacteria, is that they really only infect a certain risk group. And so typically, if you're healthy, you're not at risk. Most of the cases, about 80% of people have compromised immune systems,
Mm.
things like liver disease or diabetes, other issues. Um, Another thing that's interesting is almost all cases are in males. Females typically aren't at risk, and
Whoa.
that isn't it cool?
Yeah.
So it has likely something to do with estrogen in our bodies, as well as the fact that men typically have more iron in their blood, and Vibrio like iron a lot. It could also have something to do with the fact that men are traditionally, over the years, more likely to be out in the environment and participating in these types of activities. So
Mm.
the epidemiology side of it is really cool too.
It'll be interesting to see, as the years progress, I feel like there are more lady anglers on the water. So if that number starts to shift just based on more people participating in water activities, or if the number stays the same and ladies just have a superpower against Vibrio.
But regardless of who you are, whether you're in the risk group or not, this is my little spiel as a microbiologist. It's never a good idea to expose open wounds or tissues to any environment, not just the water. And so, if you have a cut, just put a watertight bandage over it and then just check it once in a while to make sure it's not getting wet. If you get injured while you're on the water, what you can do is as soon as you're injured, you want to get out, and if you're going out on a boat or kayaking, just bring a first aid kit with you. Otherwise, leave it in your car. And hand washing works really well, or just washing out the wound, and then just dab some rubbing alcohol in it. If you get a puncture wound from like one of those fish spines, or you step on an oyster and you get a deep cut, you can use peroxide to clean it out. Kind of gets down in there a little bit better. And then again, stick a bandage on it, keep it out of the water. It's as easy as that.
Good. That's good advice. I've definitely gotten cut by oysters more than once. So that's really excellent news that you can just wash it out and it's going to be just fine.
Usually, folks that they'll get cut or something and they won't treat it right away. Like they'll stay in the water for hours after fishing. They go home, they take a nap, they wake up 10 hours later, the infection's gone septic, and unfortunately, these infections can be fatal within about 30 hours.
Oh my gravy.
Not caught quickly can either lead to amputations, or if it gets into your bloodstream, they can be fatal in a select group of the population. But still, they're very serious things, but very easy to prevent.
Oh my goodness. Yeah, that's scary.
And I always say, they're present in any environment. So just if you take necessary precautions, treat it the same way you would like lightning or rip currents or sharks. These are all things that live or occur in the environment. You're aware of it, take the precautions, you'll be totally fine. It should not keep anybody out of the water.
That's really good advice, actually. I feel like, especially, with the shark analogy, a lot of people get afraid of the water and there's no need. You just know that it's there and it's going to be fine. Fantastic. So is there anything else that we should know about your research?
I think that's about it that I can think of. We do have a website on our Harbor Branch website specifically for Vibrio vulnificus, and it has that outreach flyer that I told you about, and then also some of the findings from our study. So you can reference folks there if they're interested in learning more.
Fantastic. I'll put that in the show notes for sure. So when I met you, you were just starting your PhD, or maybe you were a year or two into it. But you were studying shark eyes. I kind of want to touch on that, and then I want to go into the evolution of how you went from shark eyes to becoming the queen of microbiology.
They're pretty different, right?
Yes. Yes, absolutely.
Yeah.
So what got you interested in shark eyes? We'll start there because
Shark eyes. Well, I was always very interested in sharks, and as you know, Dr. Kujura's lab is very interested in sensory biology. And so that's when I started to become interested in vision, electroreception, some of the other things that they're working on there. At the time when I first started into graduate school at FAU, I was working with Dr. Tammy Frank. You remember Tammy?
I do. I took her class.
Yep. Functional biology. That was a really hard class.
Functional, but you know what? I remember a lot from that class, and maybe the harder the better. I don't know, but it was a difficult class.
It was an involved class. I guess maybe
It's
that.
very involved, and she had very high expectations.
Yes, ma'am. So I started working with Tammy, and actually, I'll say one of the great benefits to working with Tammy is that right at the time that I started in her lab, she was doing research on our Johnson Sea Link submersible, looking at
Yes.
deep sea bioluminescence. And so, one of the things that got me to work with Tammy is right about that time she asked if I wanted to come on a cruise with her, and so that was how I joined her lab, which was really
Yes, I would like to come explore the deep ocean and go play in submersibles. Thank you.
right. It was a sweet offer. So my first trip with her was out to the Bahamas to look at bioluminescence at about 3,000 feet, and I got to make 4 dives in our Johnson Sea Link submersible.
That's absolutely incredible. What
So,
was the coolest thing you saw on your dive?
wow, the coolest thing, man, we saw a huge 6 gill shark, which was just so insane and crazy to be looking out the window of a sub at 3,000 feet and see this massive shark. And they're so clumsy and they just kind of like bump into the side of the
Oh
sub.
my gosh, so you were up close and personal. It's not like he was off in the distance.
Yeah. And then on the way back up to the surface, they would turn all the lights off and all the thrusters would stir up different animals and cause them to bioluminesce. And so it was just this blue green fireworks show over the porthole window of the sub. And I just remember sitting there and thinking, this has to be the coolest thing in the world.
That's incredible. I know bioluminescence is really magical to watch. That's a really neat experience.
It was pretty cool.
So what was it like to be in 3,000 feet of water?
It's very quiet and
Mm.
very dark and it's very cold, actually.
Did you have to bundle up?
You do, yeah. So a lot of the missions that they do at the sub, were in the Caribbean and in really warm areas. And so you launch from the ship, right? And so it'd be like super hot in the middle of the summer and out in the middle of the ocean. And then as you would descend, it would get colder and colder. And the longer that you sat at the seafloor, colder it would get as well. And so you'd have to have layers,
Hmm.
keep covering yourself up. And then as you go back up to the surface, it's the reverse. It's getting warmer and warmer, and eventually you're just sitting on the surface of the water bobbing around in a million degrees waiting for the ship to pick you up. So it was a temperature extreme.
Yes. I think that's something that's kind of hard for a lot of people to grasp is that there's no air conditioning on the sub because all the power needs to go to like powering the sub.
Mm-hmm.
So, how many people are on the sub with you then?
4 in total. So the sub has 2 compartments. It has a big bubble or a sphere in the front. And
Mhm.
so that chamber has the chief pilot who is driving the sub, and then the chief scientist who is in charge of the mission. So in that case, it was Tammy. And then in the aft chamber, which is a totally separate compartment, you would have an additional pilot, and they were kind of a backup in case there was an emergency. And then the second scientist. Usually, it was a graduate student.
Okay. I remember in Tammy's class, she was telling a story about, oh no, I think it was a video of her diving in the sub, and she's leaning over the captain's chair of the sub driver, and I think she's trying to get him to grab hagfish. She,
Tammy
she
loves
like,
that.
yeah, she's like, they're slimy and disgusting, and he didn't want to grab them. And I just remember her voice like very clearly being like, but it's for science. And like I think he ended up grabbing him, which is like one of my more funny memories from her class.
That's awesome.
It's for science. Okay, so you get into Tammy's lab, you want to study shark eyes, and what's going on with your research then?
So, our original project, and so I guess the moral of the story is going to be that projects change the more you
Yes.
get into research.
Yes.
So that's not necessarily always a bad thing.
No,
Uh,
I think it's a good thing, which is why I wanted to cover it, that you can like go in and think like this is what I'm going to do because I really love sharks in your case, and then realize, a year or more in that, you know what? I don't like this. I'm going to switch it.
yep. And that there are so many different opportunities out there that I had never even known of. But so yeah, so I was working originally on deep sea sharks. So we were going to do some work as part of her cruises. And then around that time, they retired the Johnson Sealing submersible. Retired in 2010 because we no longer had our ship that was used to launch and recover it. And so we kind of lost access to some of our deep sea critters that way. So we switched our focus to near shore, and we were going to be looking at bull sharks. And we're going to be looking at their vision and how it changes as they go from juveniles that live inshore in really murky, sort of yellow green dominated waters to as they turn into adults and they go offshore into the ocean into this beautiful clear crystal blue water.
Mm-hmm.
And so that project kind of had its own challenges to it. First of all, catching bull sharks. And so I guess that would have been my first foray into fishing.
It's like, hello. Now you need to get the hook out of the shark's mouth.
Right. So here in the lagoon, we have a lot of juvenile bull sharks. It's among the largest of the bull shark nurseries on the east coast of the U.S. And so we get a lot of big mama bull sharks that come into the lagoon and they pup, usually around May and June. Actually, this is a good time to see little pups swimming around everywhere.
Oh, have fun. I'll have to go out. I didn't know that.
So we were targeting the juveniles for the first phase of the project, and so they're fairly skittish, and so it wasn't necessarily easy to get them. We weren't using gill nets or long lines for my project. We were just doing hook and line. Um,
Mm-hmm.
those were the resources that we had at the time. But so anyway, so we were catching these sharks, and it was kind of tricky. And then it was also pretty tricky to keep them alive. And so the tough thing about physiology projects is that you're trying to see how the animal's body works, right? And so it has to be working. They have to be feeding normally, so they can't be stressed. They have to be swimming normally for you to even run your experiments. And we just had such a heck of a time keeping these juvenile bull sharks alive in captivity. It was something that no one had done before. And we'd reached out to collaborators at Mote Marine Lab and the Georgia Aquarium, and everyone was like, cool, hope it works. We have no idea how to do
Oh
it.
no.
So that had its own trials and tribulations associated with it. And about a year and a half into my project, Tammy left to take a position down at Nova Southeastern, and I had the opportunity to go with her. But I loved Harbor Branch. This is where I wanted to be. I knew it was where I wanted to go to graduate school. I decided I was going to switch to another researcher up here.
Okay.
And that's when I met Peter McCarthy. Do you remember Peter?
No.
He's the microbiologist here.
Okay.
I'd known him because he, taught one of the classes in Semester by the Sea, but we never actually really talked. And I went to the summer intern symposium. So at the end of the summer, all the interns present their research. And so I was kind of on the market for a new lab and I didn't really know what I was going to do. His intern was up there presenting a microbiology project. And I just remember sitting there the whole time going, wow, microbiology is really cool. I don't know anything about it, but this sounds really cool. And so I caught him in the hallway afterwards and I said, hey, I think I'm interested in microbiology. I need a new PhD project. And he said, if you want to do something on the Indian River Lagoon, I can tell you anything you do will be novel. There have not been any projects on microbiology in the lagoon. So pick something and we'll do it. And so the first thing I did was Google fish, bacteria, and the first thing that came up was Vibrio vulnificus and that a fisherman had died in the lagoon. And I'm like, all right, that's my project. Yep.
The rest is history.
In its own weird way, it all worked out.
It really did though, because what you've contributed from the switch I feel like it's helped a lot of people. Science that, is very relatable and that people should know about, and it's fantastic.
Yeah, and that's what I really like about microbiology is I feel like that there's so many great applications to it. And, I also really like the human health side of things. My parents are both medical doctors. And so, of course, when I switched from shark eyes to microbiology, they were like, yes, we understand this. They were
Now
really,
we can chat about your work over dinner. I understand this.
they're jazzed about it too, but it's a really fascinating topic, and you know, I think it really opened my eyes to outreach too, which is the other happy part of the story, and helped me to really get involved in that, which is I think ultimately, what I really want to do.
Okay, so you graduated. I want to back up for 1 second. Did you get your master's or was it lumped in with your PhD?
Yep, it was lumped in
Okay,
for
that's
like a great big 8-year period.
okay. So it took you 8 years to get your PhD, and then the undergrad was another four on top of that, correct?
Hmm.
Okay. That's something that I want people to understand is that, just because school timelines say, oh, 4 years for undergrad, 2 years for a master's, and then 4 years for a PhD, it probably doesn't work out like that.
No way, man. It has a lot to do with the lab that you're in and the guidance that you get, from whoever you're working with. And then also your research topic has a lot to do with it. And I guess my advice would have been to younger me is to really think through the research project, and is it something that you can accomplish within, your time as a graduate student, especially if you're a master's student, 2 to 3 years, you have to make sure that what you're taking on is something that, you can really complete in that time period.
Right. So having access even to what you're studying. So in your case of the deep sea sharks, that's kind of difficult to get and requires a lot of specialized equipment and time to
Mm-hmm.
do that. So yeah, I mean, you can definitely see why the evolution happened. And then trying to raise a species of shark that's not been raised in captivity before.
Yeah. So there are a lot of questions out there, there are a lot of research questions. There are so many new things that we can do, but as a student, I think it's really important to pick a project that you can accomplish. And then I think also, who you're working with. Tammy was really awesome, but I gotta say, like, Peter is just like, as soon as I started working in his lab, it was an immediate click, so just getting along with your advisor and really being interested in what you're doing and motivated, to keep going. And I think when you start to produce findings that are just so interesting to you, that compel you, to share them with other people or to keep asking more questions, is when you really know that you're in a lab or in a research area that you really just love. And
Mhm.
that's when you're going to be successful.
You graduate with the great PhD. Now what are you doing?
So I was very lucky. I was off for a whopping 2 weeks after I graduated when I was asked to come on board as the director of outreach at Harbor Branch. I accepted the position, and what we were doing is we have a visitor center here. It's our Ocean Discovery Visitor Center, and it's a little public museum. Did you ever come here when you were?
Yes, and I'm trying to remember if I went as a student. I've been a couple of times, so I must have gone as a student, and I've been since. It's still been a long time, but yes, I have been.
Well, definitely come back. And for anyone that's listening, come to visit us. So we've totally renovated it in the last 2 years. And so we have all sorts of different exhibits that showcase the research that our scientists and engineers are doing. That's been a really cool opportunity, too, to be able to work with our researchers here and communicate what they're doing to the public. And so translating what they're doing, finding things that are cool and showcasing them, and trying to, teach people about science. And so we have this visitor center, and then we also have free tours of the visitor center, and those are done by graduate students. So as a graduate student, you can either be a teaching assistant or a research assistant to get your tuition and your stipend covered. And you should, if anyone's listening and they want to go to grad school, find a university that will give you that. Don't pay through through
Yes.
graduate school. But we started a new opportunity called a graduate student assistantship, where the grad students will work with us here at the visitor center, and they give tours and they communicate with guests and they get their tuition paid for. And the
That's
stipend.
great. It's
Yeah.
a win-win. You get knowledgeable, educated tour guides who are excited about what they're touring, what they're showing, and then they get their education paid for them.
Exactly. And experience communicating science, which no matter what your research is or what field you're in, that is so valuable.
It really is, and that's something that has come to my attention recently is that researchers are not necessarily really excellent at communicating what they're researching or what they're finding. And usually the questions that they're asking are important and have applicability beyond the scope of their lab. So it's really important to be able to effectively communicate or, partner with, a outreach center or somebody that can communicate what you're finding. So that's really awesome.
Exactly. And I think that that's something, this visitor center, it's such a unique thing to Harbor Branch. Most, we are a university here, and, most universities don't have that. This is a physical destination that is an outlet for scientists to communicate their work. And so it's a really great resource. And, over the last couple of years, we've been really just trying to optimize that. And like I said, by adding in these research themed exhibits and really trying to engage the public in all of these different efforts.
That's wonderful. Well, I will definitely make the trip up there and check it out. What is your coolest project that you've worked on? Or your favorite field story to tell? And it could be like, I can't believe that happened day, or just like that was so epic day.
Okay. So it's another sub story. So
Okay.
I think the sub just by default has to be like the coolest thing ever.
Right. I mean, yes, absolutely.
So
That's pretty hard to top.
yeah. So the work that we were doing in the Bahamas was really cool, but my most memorable story, um, the very last research cruise that the Johnson Sea Link submersible went on was a response to the oil spill in the Gulf of Mexico. And so right after the oil spill happened, they mobilized a team and they sent the ship and the sub over to, West Florida. And we were documenting some of the deep reefs there to try to see, what was there before in case the oil hit it so that we could go back and compare the damages.
Right.
And so we were working, I think it was just north of Key West at this point. We were out for almost a month at a time. And we kind of worked all around West Florida, and then we looped around and came back up on the east side. But I think we were around Key West, and we were diving down to about 1,800 feet. Tammy was in the front. And so in the front, they have the big sphere so they can see everything that's going on around them.
Okay.
And then the back has a little video screen. And so you can see the feed from the camera that's up front. And then you set so you can communicate. And so Tammy shouted, "Oh, look, a swordfish." And so I like go to my little cubby window thing and look out the window and looking, and I don't see anything. So we keep going on, and a few minutes later, this huge swordfish slammed into the window on the sub.
Oh my gosh.
Yeah. So it had seen the light above the little porthole.
Okay.
And I guess it went to hit at it because usually it's pitch black down there.
Oh, so it was like, I'll see you.
Yep, I'm gonna eat you.
Oh my gosh.
But it didn't work because its bill actually got stuck.
Oh my gosh.
In the side of the sub. So I saw it happen, and so usually the sub pilot in the front is the one driving, and the person in the back is just backup. So like usually they like put on headphones, they like read a book or like take a nap or whatever. So the guy was like reading a book quietly, curled up in the corner. I tapped him on the shoulder, and "Excuse me, there's a swordfish stuck in the sub."
You might need to go to work now.
So he looked, and sure enough, it was stuck. So unfortunately for the swordfish, all the arms on the sub face forward, so there was nothing on the side. So it didn't make it, but we took this thing around with us for the rest of our dive and wound up surfacing with it. And everyone on the boat got to eat swordfish that night. I don't eat fish, but they were they were all very excited for it. So.
Oh my gosh.
I ever caught.
I feel kind of sad for the swordfish, I'm not gonna lie, but that is a hilarious story.
It was legal size. That's what everyone kept saying. We were all like feeling so bad for it. And they're like, "It was legal."
It was legal. It was legal size.
Things. I'm like, "Oh my God, did it hit like a wire? Is it good?" We're like at the bottom of the ocean. We're dangling this fish.
Wow. And you said you were at 1,800 feet?
Yeah.
Wow. I had no idea that they went that deep. That's pretty impressive.
I think that's where they hang out. And then they may come up closer at night to feed on certain things that migrate. I'm not sure, but yeah,
Okay. Okay. I guess that does make sense.
I don't know what you'll see down there.
Right. It's just a whole nother world. So, what is your favorite part about your current job?
Ooh, that's a good question too. I love this current position, and I think I've always really gravitated towards education and outreach. So, subconsciously, it was probably something that I always wanted to do. So it's really cool that I'm doing it now. But I really love translating science to the public. I think that not a lot of people do it, and like you said earlier, do it well.
Mhm.
And so I like, meeting with researchers and hearing their story and seeing what they do, and then taking that and kind of packaging it into something that the general public can find interesting and relatable. And yeah, and just engage them and get them to want to come back and learn more and go to lectures and maybe encourage people to become marine scientists. We get a lot of students that are on the fence about marine science, and at the visitor center we have 5 different exhibits, and each one is a different research area at Harbor Branch. And I always think it's so cool that when people go on these tours, they get to see that there's so many different things that you can do as a marine scientist. You're not just swimming with dolphins or corals, like you're creating new medicines from deep sea sponges that can cure cancer.
Mhm.
You're creating sustainable seafood through aquaculture, and you're building these underwater robots and equipment that can help us survey the ocean. So there's just so many different topics, and I just love sharing that with people and opening their eyes to marine science.
Yes. Yes, absolutely. There's just so much depth to the field that people don't realize, which is really why I started this podcast to begin with.
Yeah, it's a great idea.
So, what's your most frequently asked question? And that we could take it back to your PhD research or at the visitor center.
Hmm, that's a good one too. I guess the most frequently asked question would probably be about Vibrio,
Okay.
because I did so many lectures and interviews and things during that research, and just people wanting to know, what it is and how they can become infected, or most importantly, how they can avoid infection.
Right.
And it's such a big health concern, for sure.
Absolutely. Yeah, because people want to be able to go and play in the water and not worry about getting a flesh-eating bacteria.
Yeah, exactly.
It's
And
understandable.
usually you hear more about them in the summer than in the winter. So they do have that slight seasonal correlation where they're more abundant in the summer when it's warmer, even though they are here year round. But
Mhm.
I actually just started seeing over the last 2 weeks, they're starting to pop up in the headlines. And so I suspect we'll be getting a lot more inquiries over the next few months.
Makes sense.
Folks might be seeing stories about them in the paper, and you can
All
say
right.
about them here.
Keep keep my eye out. So, do you have, or what advice do you have for aspiring marine biologists? And is there any advice that they should ignore?
Ooh, okay. Well, I'll give them advice first. So, I guess if my story is told anything, it's to be open to new experiences, and to getting as much experience as you can. I think that's the most important thing, is that, all of our lives, while we're going through school, we're told that we just need, good grades and to study and
Mhm.
get good grades. But with this field, you really need experience. And so at, a young age, if you're interested in marine science, you should be trying to volunteer, at a nature center, an aquarium, just try to get some experience. And then as you go through school, try to get in with a lab. And you know what? It might not necessarily be in the research area that you're going to eventually work in, but just some experience. And that's
Mhm.
a good way to find out if you like what you're doing.
Mhm.
you know After I was in semester by the sea, I went on to teach semester by the sea for 6 years. I taught marine biodiversity and the microbiology course.
Mhm.
And there were several years where we would get students to come to me halfway through the semester and say, you know what, I didn't think I thought I would like this and I don't, and I want to change careers. And I'm like, great, this is when you find that out,
Right.
It's okay. Like, learn about what you want to do, and it's an experience in your life.
Right.
So I think just get as many opportunities and experiences as you can, and hands-on, and meet people, because in this field it is all who you know. It is connections.
It really is. And it's amazing how small such a big field can be.
Right?
Like there's 6 degrees of separation in the world, supposedly. I'm going to go with marine bio cuts that in half. So as we wrap up, one of my favorite questions to ask is, what's your favorite sea creature?
Oh, okay. Well, hands down. So my favorite sea creature and probably animal in the world is a whale shark.
Mm.
They're the most amazing animals in the world.
Why do you love them so much? I mean, they're incredible, absolutely, but do you have like specific reasons?
I like their pattern. I think that's what's always done it for me. They're just so unique. Like, how can this animal that is so huge have this just amazing, color pattern that's unique to individuals? And they're amazing.
They really are. Have you swam with them?
I have. Oh gosh, many years ago now. I actually went to Mexico, to Isla Mujeres, during the aggregation, and we swam with about 200 whale sharks.
Oh, that's absolutely incredible. That's on my list, that I want to do that.
It was just so cool. I cried.
Is there anything else that you want to add? Any last pearls of wisdom?
Oh, pearls of wisdom. I think that's it. Come visit us. Tell your guests to come visit us here at Harbor Ranch, learn about science. I'm here at the visitor center. I'm happy to, give people tours and tell them more about what we do here. And, if you're not near here and there is a local science center or research institute, check them out online and see what kind of outreach programs they have, because now, more than ever, these types of institutes are realizing the importance of communicating science, and they're investing in efforts that try to connect people with scientists. And so use those resources as an opportunity to meet scientists and ask them questions and learn about what they do and, what path they took to get into their position.
Absolutely. Well, thank you so much for being on the podcast, Gabby.
Thank you for having me.
Thanks so much for listening to today's episode. Now I'm off to go visit Gabby at Harbor Branch's Visitor Center. And that is my challenge for you today. Is there a nature center by you that you could go visit? Even if you don't live on the coast, getting out and learning more about the wildlife in your area, the environment in your area, it's always a treat and a great way to spend the day. And I guarantee you will learn something new. I always do. So get out there, y'all. I'll talk to you soon. Thank you for listening to today's episode. If you enjoyed it, I'd love to hear from you. Leave a comment under the show notes at marinebio.life or send me an email at hello@marinebio.life. We'll catch you next time on So You Want to Be a Marine Biologist.